Hey guys, welcome back to my channel Deep Biology. In this video we will discuss the question from December 2019 CSIR Life Science exam. The question is, select a cellular body which is not a part of nuclear body. We have to find the cellular body which is not found in the nucleus. The second option is nucleolus. The nucleolus is found in the nucleus, which synthesizes the rRNA protein, which will transport in the cytoplasm and leads to the synthesis of ribosome. The third one is Kajal body. The Kajal body is found in the nucleus, which is responsible for the splicing of mRNA. That's why second and third cannot be the answer. The fourth one is interchromatin granule cluster. The interchromatin granule cluster are also found in the nucleus, which have factors which are responsible for the pre-mRNA splicing. That's why fourth can not be our answer, and our answer is P bodies, because P bodies are not found in the nucleus, they are found in the sieve tube of phloem in angiosperm plant. It is not found in the nucleus of the sieve tube. That's why our answer is P bodies. Now let's move to the next question. The next question is, one of the mechanisms used by bacteria for adaptation to changed environment is altering transcription of their genes. In this regard, which one of the following is not found in the bacteria? We have to find that in addition to changed environment is altering transcription of their genes, we have to find the statement which is not found in the bacteria in respect to the alteration of transcription of their genes. First option is a gene with two different promoter for expression in different condition. This is the right statement, because the two different promoter can be used for expression of same gene. For example, when the gene is expressed in root and when gene is expressed in leaf, there is found two different promoter. That's why one cannot be our answer. The second one is used of different sigma factor for transcription of genes. In different environmental responses, there are found different sigma factor for transcription of genes. That's why second cannot be our answer. Expression of anti-sigma factor is also a right statement, because the anti-sigma factor such as inhibitor will inhibit the action of sigma factor. That's why the RNA polymerase cannot found in functional form, and there will be not found any transcription. Fourth is also a right statement and cannot be our answer. The third one is our answer, because expression of alternate of alpha beta and beta dash subunit is a wrong statement. There are found same alpha beta and beta dash subunit in all RNA polymerase found in an organism. That's why third is the right answer. We will upload video on CSIR Net Life Science question on regular basis. So please like, subscribe and share with your friends for enhancing your knowledge. Thank you and have a nice day.